Hello everyone. This voice thread will have two sections. The first one is going to be on data management life cycle, and the second one is going to be on the different aspects of biostatistics. This is the overall lesson learning objectives for the lesson. As an outline of this lesson, first we're going to have background discussion on clinical data management and clinical data management systems, or CDMS. CDMS are the softwares that we use as a database. And then we look at the data management standards and guidelines. These are what shapes our data management process. And then we look at the data management process, starting from developments of data management plan up to database closure and archival. Clinical data management is a process of collecting, cleaning, and managing of a data in compliance with the standards. And the primary objective of clinical data management is to provide high quality data, which is ready for statistical analysis. And what we mean by high quality data is it has to be accurate and suitable for statistical analysis. And it has to also meet protocol specified parameters and comply with the various protocol requirements, and also have minimal or no misses. As you know, this is one of the headaches in clinical trial, and it's going to be the source of bias. And finally, have acceptable level of variation, which will not affect conclusion. Then we move to clinical data management systems, or CDMS. CDMS are the softwares that we use as a database for storing data. And they could be either commercial or free. And examples of commercial CDMS systems include Oracle Clinical, ClinTrial, eClinical Suite, Macro, and Rave. And from the free types of CDMS, we have got the famous OpenClinica, OpenCDMS, TrialDB, and Fosco. RedCap is also a free types of CDMS, but we need to get permission from the university responsible for building it. The bare minimum requirements of CDMS by regulatory authorities is that it needs to be password protected with individual access, and sharing of passwords and usernames is not going to be allowed. There should be audit trail, meaning the initial entry and the subsequent entries should be visible or recorded somewhere. And the third one is there should be date and timestamp for us to be able to understand when was the initial entry, when it was changed, and also who has changed it is going to be documented in the audit trail. The other aspect is on standards that we use in data management. So there are various standards guiding the data management process, and these standards include even guiding us on how to standardize names, codes for variables, and also the structure of the data and the formats across locations, studies, and organizations. So some of the benefits of the standards in data management include cost-effective and timely product development. So as you know, at the end of phase three trials, we are going to submit the data to regulatory authorities, and these authorities, they have to go through in detail the data submitted to them, and they have to make a decision whether marketing authorization has to be given to the sponsor or not. So most of the time in the past, what happens was that the regulatory staff, they will spend too much time organizing and reorganizing the data so that they can understand the results better. So using standards will facilitate the review process by the regulatory staff, and hence that means we're going to have timely product development. The second benefit of standardization is it will streamline the process for conduct, data transfer, analysis, and submissions. The third benefit is it will decrease the time and money needed to set up a new study. For example, if an organization has already conducted a clinical trial in the past, and if that organization is already using standards, then setting up similar studies is going to be easier because of the existing standards. Finally, comparison of data across sites and data sharing is going to be utilized to its maximum potential because of data standards. Various organizations and consortia develop and also maintain the guidelines for data management. And this includes the Clinical Data Interchange Standardization Consortium, or CDISC for short. As you know, FDA has got also different standards and guidelines. ISH is another source of these standards and guidelines, and we have got also the Society for Clinical Data Management, giving rise to various standards and guidelines. So the first guideline commonly mentioned is 21 CFR Part 11. This is a Code of Federal Regulation, and this is mainly on electronic records. So our database has to be compliant with this regulation, and also if we are using electronic CRF, it has to be also compliant with this. The other guidelines include the Clinical Data Acquisition Standard Harmonization, or CDASH for short. The CRFs that we develop has to be compliant with this requirement. The other is Standard Tabulation Data Module. This is mainly for data presentation, and we have got also ADAM, which is Analysis Data Module, and this is for statistical analysis. And all of these three guidelines are coming from CDISC. We have got also good clinical data management principles, which is a very comprehensive guideline, and it gives guidance on how to operate in terms of data management, and that's coming from the Society for Clinical Data Management. Next, we discuss about data management process, and we start the process with review and finalization of the study documents. We review the protocol for visits, events, and data to be collected in the study. Particularly, we will refer to a table in the protocol called Schedule of Events, or the other name to it is Visit Matrix. And this is a table which indicates the different types of visits in the study, and also the types of events and data which are going to be collected in each visit. Following that, we developed the data management plan. This is a comprehensive plan detailing the activities in the data management process. And some of the things that could be included in data management plan includes the CDMS that we're going to use, including its version, the various types of activities are linked with persons who are responsible for it, and other details are included in the data management plan. Apart from the data management plan, we also developed the data validation plan or data cleaning plan, and this plan indicates the various critical data that are going to be cleaned and the manner in which they are going to be cleaned. So possible way of cleaning the data includes using edit checks, which are embedded in the database, or extracting the data from the database and using external tools to clean them. Or the other option could be just manual checks on the data listing. So if the sites pay particular attention on the data that are indicated in the data validation plan, probably they're going to be more productive. The second step is developing the CRFs, and this is one of the critical steps in ensuring quality data. At earlier stages, probably the first important thing to do is to design carefully the source documentation. And from the source documentation, this will be followed by designing the case report form. And together with the CRF completion guideline, these three aspects are going to be really critical in ensuring data quality and also minimizing the data queries that are going to be generated downstream. In the previous slides, we've seen that the data management process starts with reviewing the study documents, followed by developing the data management plan, including the data validation plan, and then we develop the CRF. Then the next step will be to design the database, and this has to be done in compliance with regulatory requirements. And as we've seen previously, the bare minimum requirements is that the database has to be password protected with individual access, and also there should be audit trail, and also date and time stamps should be there. After developing the database, we need to validate the database, and what we mean by that is that we need to have a system specification, we need to have user requirement tests, and also we need to ensure regulatory compliance by running some tests and documenting the results of these tests. We need to have test environment to check on the different functionalities of the database. And once we are satisfied, then we have to release the database for production. And in production environment, then we can start the actual clinical trial data collection process. Now, one of the common challenges for sites which are handling the data management process in-house is that the database validation aspect is usually overlooked, and sites which are overtaking handling this process, this responsibility, they need to make sure that their systems, their CDMS, are validated. The next step is data collection, and we can have either paper-based studies or electronic studies. And in case of paper studies, we have both paper CRF, and there is more transcription in such a way that the data is going to be transferred from the source document into the paper CRF, and from the paper CRF, data is going to be entered into the database. Whereas for electronic studies, which uses electronic CRFs, the data is going to be entered from the source document directly into the database. Now, the other aspect is that electronic studies, they are with less errors because of minimal transcription compared to the paper studies. And there is also faster resolution of discrepancies for electronic studies. This is because in the case of paper studies, the paper CRFs are going to be sent to the data management units. And the data management units, when they start entering the paper CRFs into the database, they will learn that there are some discrepancies and errors. And then they have to put or print these findings on a paper and send them to the sites for resolution as data clarification forms. Now the sites will work on resolution of these discrepancies, and finally they will send back the resolution to the data management unit, and this takes time. Whereas in the case of electronic studies, since the sites are entering the data directly into the database, they will directly look at the discrepancies and errors, and they will make the corrections there and then. So that way, the resolution of discrepancy is faster for electronic studies. The other point worth mentioning is that any data that's entered into the electronic CRF by the sites should have a PDF of the paper, the PDF of each screen of the electronic CRF to be filed on their sites, because it may so happen that some of the data that they are entering, they don't have any source, and hence they have to keep all the screens as a PDF. Then the next process is CRF tracking, and CRFs are monitored before shipment to the CDM, and the data management unit also, upon receipt of the CRFs, they will check for the completeness of the CRFs, and this usually applies for paper studies. So the data management unit will check on gross problems on the CRF, such as missing pages or illegible types of data. And if there are any such issues, the data management unit will send for clarifications to or corrections to the sites, and the sites will respond to that. Then the next step will be data entry. So we can have single data entry or double data entry, and double data entry is commonly practiced. And in the case of double data entry, we will have two people entering the data. And in terms of discrepancies between the first and the second data entry, one option is that the second person is going to be more experienced, and that person can make the corrections. That's one option. Or the other option, we're going to put the third person as a chain breaker. So if there are any discrepancies, the second person is not going to make the corrections this time, but the third person will be able to do that. So data entry could be either paper-based or it could be electronic data capture, which means in the case of electronic data capture, it's going to be, we're going to directly enter the data into the database. The next step will be data validation or data cleaning. So basically, this is testing the validity of the data with protocol specification. And as I've indicated earlier, three options are there for the data validation. The first one is using edit checks. The second one is extracting the data and using external tools to clean the data. Or the third one will be manual checking using the data listings. So regarding edit checks, the edit checks could be the front end edit checks types. This is the most common one where you get to see the errors right away, when you enter the data. Or sometimes the edit checks could be back end edit checks, which means the checking is going to be fired only once all the data is entered. So edit checks that fail to pass validation are called discrepancies. And this could be inconsistent data or missing data in case if the field is a forced one, or it would be error related to range checks. For example, you can't have a person who is 10 meters, or it could be a protocol deviation. So discrepancies could be any of these forms. The discrepancies are going to be seen, as we've discussed previously, for the case of electronic CRF right away when the sites are entering the data. But in the case of paper CRFs, the sites are not going to look at the discrepancies right away, but instead, the sites are going to get the discrepancies in the case of paper CRFs as data clarification forms after the data management unit has printed the queries or discrepancies on a paper. The other aspect is discrepancy management, also known as query management. This is a very critical process in ensuring the data quality. So to manage the discrepancies or queries, we need to review the discrepancy, investigating the reason for the discrepancy, resolving them with documentation, or declare them unresolvable. So the CDMS at the database should have a dedicated section to record all those discrepancies that has been observed in the database, and also including their status, whether they are resolved or not. So at the end of the study, when the database is closed, we need to have all the discrepancies closed. Otherwise, closing the database without closing the discrepancies is not going to be acceptable. So the discrepancies are going to be either flagged for the investigator to respond to, or most of the time we can have also self-evident corrections, meaning these are the kinds of errors and discrepancies that can be automatically corrected by the data management unit. But here we need to emphasize that changes made by the data management unit, which is designated by the sponsor, is not something easy, and hence for self-evident corrections to take place, either the sites have to send a list of possible self-evident corrections that could be corrected by the data management unit at the beginning of the study, or the data management unit has to send all such self-evident corrections for approval to the sites, or combination of both could be possible. Otherwise, without such process, the data management unit cannot make any change on the data submitted by the sites. Most of the time, the data that we collect in clinical trials are very much structured. But sometimes we have got also open-ended questions in conditions such as safety issues, conditions such as medications, medical history, and the likes. So in such conditions of open-ended questions, we need to code the responses given by the sites or by the patients. And hence we need medical coding. So medical coding uses standard terminologies for conditions like adverse events, medications, medical history. We have got MEDRA, which is a medical dictionary for regulatory authorities, mainly used for coding adverse events and also medical histories and others. And we have got also the WHO drug dictionary enhanced, mainly used for coding medications. So if we have got wrong coding, then this will result into masking safety issues, or wrong safety concerns or signals are going to be detected. But medical coding, if used properly, is going to be useful for acute reporting of safety, efficient review, and also data sharing. In case if you are wondering what are the qualities required by medical coders, it includes knowledge of medical terminologies. It also includes understanding of the disease entities and pathological process. They need also to have understanding of the drugs, and most importantly, the coders should have knowledge on the structure of the electronic medical dictionaries and the hierarchies in it. At the end of the data management process, the database is going to be locked, and this is going to be done after final quality checks and assurance has been done, and also after final validation check. And basically, all the data management activities should be finalized, including discrepancies. So the discrepancies, they have to be either resolved or they have to be closed somehow. And the standard procedure is to use a pre-log checklist, and this checklist indicates all the possible activities that needs to be done before the database is logged. And again, in this checklist, we are going to indicate who is going to be responsible and by what time it's going to be conducted. And finally, whenever the activities are done, then the responsible person is going to sign them off. So signing will be important. So we follow this strict rule because unlocking the database after it has been locked is something which is frowned upon. So it could be an indication of fraud. So unless you have got really convincing reason, you can't unlock the already locked database. Additional point here is that the use of checklists is very much helpful in clinical trials. So this will make sure that important steps are not missed, and hence for situations such as database lock and also initiation of the study, we can use initiation checklist. And for site closeouts also, we can use site closeout checklists. So this will tremendously improve the quality of the conduct of the clinical trial. After the conclusion of the study, the data has to be archived, and this is both after the successful completion of the study or even discontinuation of the trial. All the data has to be archived, and ISA GCP recommends that the data has to be archived for 2 years. And there may be also other stringent requirements. Some of the pharmaceutical companies require the archival for something like 15 years or 20 years. So you have to check with your sponsors for how long the materials, the data is going to be, the essential documents are going to be archived. What is the importance of the archival? First, it's to meet the regulatory requirements. The second one is to respond to questions that may arise long after the trial has been concluded or discontinued. The other aspect is when you archive the data, it has to be done in such a way that you can easily retrieve the required data within a reasonable time frame. So this is a responsibility of the sites, and probably this is something that needs to be also reflected in the clinical trial agreement with the sponsor. And the sites, they should have a facility to archive the data, or otherwise the other option is they need to find other vendors which can do that for them. The second part of this voice trail is going to be on discussion of sample size in clinical trials, hypothesis testing, and bias in clinical trials. These are the overall lesson learning objectives. As an outline of this lesson, first we're going to discuss sample size in clinical trials, and then we're going to discuss different types of errors in biostatistics, and particularly in relation with sample size and power. And then we're going to continue our discussion with concepts of hypothesis testing. And then we continue with sensitivity testing. And finally, we conclude with bias in clinical trials. When we conduct clinical trial in a particular population, for that matter, when we do any type of clinical research, we can't study the whole population for reasons of logistics and cost issues. And that means we need to take a sample population to make study and make inference on the target population. So we need to have an optimal sample size. And this sample size, if it is too small, then the difference between the arms to be studied in the population in the study could not be detected. Or if the sample size is too large, then again the cost and logistic issues applies. So when we calculate sample size, there are some factors which we need to consider. And the first one is going to be type 1 error or alpha. And the second one is type 2 error or beta. And the third one is minimal clinically relevant difference, or the other name for it is effect size. And particularly for continuous types of data, we also factor in the variability. Before we look in detail about the factors that we mentioned in the previous slides affecting sample size, it will be necessary to look at the concept of hypothesis in statistics. So in statistics, we have got null hypothesis and alternative hypothesis. The null hypothesis, denoted by HO, states that there is no difference between the groups, and the alternative hypothesis, denoted by H1, states that there is actually a difference between the groups studied in the clinical trial. So after doing a statistical analysis, we may either reject HO if there is a statistically significant difference, or we may accept the null hypothesis if there is no statistically significant difference. Now, with this concept in mind, now we can now discuss about the different types of errors in statistics. The first statistical error to be discussed is type 1 error, or alpha. So this is an error which occurs when we wrongly reject the null hypothesis. So rejecting null hypothesis means an association, and hence this is a type of error that gives you false positive association. So alpha is the probability of rejecting the null hypothesis where the null hypothesis is true. And normally we take this margin as 0.05 or 5%. And here what we are saying is that, for example, if we get a p-value of less than 0.05, then you are saying that the probability of finding this result by chance is less than 1 in 20. And the other aspect is that statistically significant findings doesn't mean that they are absolutely free of chance occurrence. Because, for example, if you have got a p-value of 0.03, then still there is 3% probability that the finding is by chance. So if we decide to reduce the alpha value smaller than 0.05, then the sample size is going to be very much huge. The second type of error in statistics is type 2 error, and it's also called beta. And this is a type of error when we wrongly accept the null hypothesis. So we said accepting the null hypothesis, we are declaring that there is no association, and hence this is a type of error which gives you false negative association. So beta is the probability of accepting the null hypothesis where the null hypothesis is not true. And for many studies, we take beta as 0.2 or 20%. But in phase 3, usually this error is reduced to 0.1. And the other aspect is power is complementary to beta or type 2 error in such a way that power is equal to 1 minus beta or type 2 error. And when we define power, it is the probability of correctly rejecting the null hypothesis where it's not true. And if we decide to increase power, in other words, to reduce type 2 error, then the sample size is going to be increased. The third factor determining sample size is minimal clinical relevant difference or effect size. So this is a difference that the investigator believes to be clinically relevant and also biologically plausible. For binary variables, the difference or the effect size is expressed in percentage. For example, a difference of 10% between the intervention and control group. And for continuous data, the difference or the effect size is expressed in numerical figures. For example, if you are talking of an intervention that will have an effect on blood pressure, then you can talk of a difference of 15 millimeter of mercury systolic blood pressure between the intervention and control group. If you are aiming for effect size, which is very much small, then you need a very huge sample size to detect this effect size. And to the contrary, if you have got a relatively large effect size, then the sample size required is going to be relatively smaller. The other factor determining sample size is variability, and this applies to continuous variables, which compares means of study population. So usually variability is expressed as standard deviation or variance. And usually when we calculate sample size, we obtain this variance from pilot studies or previous similar studies. And what happens is that if the variance is huge, then the sample size is going to be very huge. Apart from the four main factors affecting sample size, we can have also other factors influencing sample size calculation. The first one, for example, could be expected distribution, whether the distribution is normal, binary, or ordinal, could affect the estimated sample size. The second one is the type of comparison that we are aiming to make between the groups. whether we are aiming for superiority, non-inferiority, or equivalence types of comparison, again, could influence the sample size. And the type of design, whether it's a parallel design, crossover design, or multi-arm, again, could affect the sample size. Now that we've looked at the factors affecting sample size, then we move to hypothesis testing. So most trials, they will compare treatments, particularly at least phase 2B or 3 trials. They will be comparing the active intervention against placebo or standard of care. And superiority trials are the types of comparisons which are very much common, and they are also consistent with the hypothesis testing logic, and they are aiming to show that a particular treatment is superior to another one. And the type of statistical testing depends on the type of data. For example, for continuous types of data, usually we aim for comparison of means, and t-test, ANOVA, and the likes are going to be more convenient. And for categorical data, we're going to compare proportions of the different groups or arms. And well-suited tests include chi-square tests and the likes. And yet for the time to event types of data, we're going to compare the hazards of occurrence in the different groups, and we're going to calculate hazard ratio. And for that, we may use tests such as a log rank test or Cox proportional method. In this slide and the subsequent slides, we'll look at the logic behind hypothesis testing. And hypothesis tests assume that the two groups which are compared are equal under null hypothesis. And the objective is to demonstrate that the data is extreme or rare. And two possibilities are there after the hypothesis testing. The first one is the assumption that we made under null hypothesis is false, which is there is association. Or the other possibility is that the extreme data that was observed was by sheer chance, and which means you don't have any association. Statistical theory sets a threshold to define extreme results as rare. And these thresholds are set based on our willingness to draw a false conclusion. And usually, our willingness will be to draw a false conclusion 1 in 20 or less than that, which means our level of significance or alpha value is usually 0.05. And if the p-value is less than the margin or the threshold that we've set, meaning p less than 0.05, then we conclude that the assumption under the null hypothesis is false, which means we reject the null hypothesis and we declare association. So as we've said, if the assumption under the null hypothesis is false, then that means the two groups are not equal, they are different. But still there is a 5% chance that this conclusion could be incorrect based on the alpha value that we said. And that means for an alpha value of 5%, 1 in 20 times our conclusion could be wrong. If the p-value is greater than 0.5, then there is lack of evidence of a difference. But still, when we increase the sample size, the association may appear. Determination of association will be using distribution curves, particularly so for parametric types of tests. In statistics, we have got parametric and non-parametric types of tests. And the parametric types of tests, they are very much dependent on some assumptions, and the non-parametric ones are a bit relaxed in terms of assumptions. So curves such as normal curves, binomial curves, Poisson, chi-Weibull distributions could be there as curves. And the curves may be having different types of shapes, but what we aim to do is to show the results that we've got is very extreme, very rare. So as you've said, whatever the curve, whatever the test that we use in statistics, the final goal is to show that the result we obtain is very rare and located at the extreme end of the tail where the probability is less than 0.5. So in the subsequent slides, we're going to look at what this means. This is a normal standard distribution curve with a mean of 0 and a standard deviation of 1. The area under the curve is 100%, and the area between the various standard deviations are indicated in this slide. For example, 68% of the area is between the standard deviation of -1 and 1, and 95% of the area is between the standard deviation of -1.96 and positive 1.96. And yet 99% of the area is between the standard deviation of -2.58 and positive 2.58. Now, when we say our margin of error or alpha value for our study is 5%, we are setting our margin at 1.96 standard deviation. So we collect our data, we analyze our data, and we look at our calculated statistics. And if our calculated statistics falls at the extreme end of our margin, then we reject the null hypothesis and declare association. But to the contrary, if the statistics that we have calculated lies in the middle, then we are going to accept the null hypothesis. The other way of putting it is we calculate our statistics and we compare it with the tabulated statistics. And if the calculated statistics is on the extreme end, then we reject the null hypothesis. This is a t-test distribution, and the story is the same. If your calculated statistics lies on the extreme end, then you reject the null hypothesis. In this slide, you see various types of distribution, including chi-square test distribution, binomial test distribution, Poisson distribution. And in all of the cases, if your calculated statistics is on the extreme ends, then you reject the null hypothesis and you declare association. So additional curves are indicated here. Here you have got the F distribution, the linear regression distribution, and the log rank test distribution. And in all of the cases, you reject the null hypothesis when your calculated statistics is on the extreme. The other aspect is about selection of a statistical test for the data that we have collected. And this depends on several issues. For example, the type of data that we've collected, whether it's continuous, categorical, or time to event types of data, will decide whether we need to select a particular statistical test. And the expected underlying distribution will also be expected to influence our statistical test. There are several distributions, as we've seen in the previous slides. And the other factor is sometimes authors have used a particular statistical test for their publications, and we tend to follow these previous publications, previous articles, the way they have analyzed their data. And we have got also international recommendations on how a particular data should be analyzed. And we also need to pay attention to these international recommendations. And finally, we have got parametric and non-parametric types of tests. And in parametric tests, there are several assumptions made. And if our data is not compliant with these assumptions, then we go to non-parametric types of tests, which doesn't make much assumptions. And the disadvantage of non-parametric tests is that you will lose power by using them. The other issue is about sensitivity analysis. Sensitivity analysis simply means you analyze your data in different ways using different statistical tests and using different assumptions. And if your result is one and the same, then you have got a robust data or robust result. So sensitivity analysis is a method to check robustness of a result. And it's especially important for confirmatory types of trials where marketing authorizations are going to depend upon it. So types of sensitivity analysis could be used, could be done, including or excluding outliers. If you have got similar result, your data or your result is robust. And the other one is assuming assumptions on non-compliance and protocol violation. So you do per protocol or intention to treat analysis. Both of them, they have to be done for superiority comparison types of trials. And based on both per protocol and intention to treat, if you have got the same result, then you have got a robust data. The other one is assumption on missing data, whether including or excluding missing data gives you the same result, and your data is robust. The other aspect is based on definitions of outcomes. Outcomes could be defined in different ways, and these different definitions, if it gives the same result, then your result is again robust. You have seen different types of distributions in the previous slides, and assuming these different distributions, if you get similar result, then again your result is robust. The other one is baseline imbalance, adjusting or unadjusted results. If it gives you the same result, again, your data is robust. These are the different examples of sensitivity analysis and the scenarios. For example, for non-compliance or protocol violation, we may use intention to treat, or as treated analysis or per protocol analysis. And for missing data, we can analyze only complete cases, or we can impute the missing data and we can include in the analysis. For the different definitions of outcomes, we can include different cutoffs or definitions and analyze the data. And for the distributional assumptions, we can perform the analysis under different distributional assumptions. For example, using Poisson distribution, negative binomial distribution, and the likes. Or it could be parametric and non-parametric methods of analysis, classical versus Bayesian, and the likes. Bias in clinical trials are very much critical in deciding the validity of the result coming out of our trials. And hence, we need to have procedures in place to avoid bias or to minimize them. We have got at least 5 types of bias in clinical trials, from starting including conduct and also reporting of the trials. These bias include bias arising from the randomization process, also called selection bias, bias due to deviations from intended interventions, also called performance bias, and bias due to missing outcome data, also called attrition bias, bias in measurement of the outcome, also called detection bias, and finally, bias in selection of the reported result, also called reporting bias. So here we'll be dealing with the first four bias, and the fifth one will be dealt with in lesson 7 when we discuss about closeout and reporting of clinical trials. The first bias to be discussed is bias arising from the randomization process, and this is also called selection bias. And in such kinds of bias, we're going to have either poor or good prognostic factors more distributed in either of the arms. And the idea is to prevent these types of excess distribution in either of the arms. To prevent these types of bias, randomization is employed. And randomization is meant to ensure equal distribution of known and unknown prognostic factors between the different arms or among the various arms. And critical process in randomization includes sequence generation. And usually what's recommended is to use computer system for generating the random numbers. And the second one is concealment of these sequence generated. And for concealment, we use opaque envelopes to prevent people from looking at, the assignment. And also the other option is to use IV arrays or interactive voice response systems, or IW arrays or interactive wave response systems. The second bias to be discussed is bias due to deviation from intended interventions. This is also called a performance bias, and this is due to favorable treatment of participants assigned to the intervention group with the hope of getting positive results. And this could be intentional or unintentional. And the way to prevent performance bias is by ensuring blinding of the staff involved in the process. And this will reduce or avoid such types of bias. The other bias is bias due to missing outcome data, also known as attrition bias. And what happens is that usually missing data is biased towards participants showing unfavorable intervention effect, which means if a participant shows unfavorable intervention effect, uh, there is a tendency to for that data to be missing intentionally. And what's ideal is if no data is missed in the trial, and to make sure that such bias didn't occur in the clinical trial, we do both intention to treat analysis and also per protocol or according to protocol analysis. And in per protocol analysis, we only take data from participants who are almost fully adherent. And in the case of intention to treat, we take data from almost everybody, and we tend to include everybody as they are randomized at the beginning. Now, in the intention to treat, since the data collected is including participants who are not fully adherent to the administration of the intervention, it may undermine the intervention effect. Whereas the per protocol or according to protocol analysis, since it only selects individuals, participants who are fully adherent to the administration of intervention and other procedures, it kind of overestimates the intervention effect. So the take-home message here is that sites need to plan to minimize or avoid missing data, because if you have got too many missing data, then the results that we get for the intervention is going to be undermined, and that's going to be to the disadvantage of the sponsor and ourselves. The last bias to be considered here is bias in measurement of the outcome. It's also called detection bias. This is as a result of difference between groups in how outcomes are determined for them. When a staff is aware of the treatment allocation, then that staff may treat differently the different arms depending on the assignment. That means um, if bad outcome is expected from the two arms or from the different arms, the outcome which is going to be reported for the ones assigned to the intervention group is going to be minimal. These types of bias is going to also depend on whether the outcome is objective or subjective. And if the outcome is subjective, then we're going to have lots of problems. But if it's objective, then the damage is going to be minimal. So blinding is important to avoid or reduce such types of bias.